How our immune system, subunit vaccines made with recombinant DNA. Have you ever wondered how some vaccines can train your immune system without using the whole virus or bacteria? Today, we will uncover the fascinating process behind subunit vaccines made with recombinant DNA. Subunit vaccines focus on specific parts of a pathogen, usually proteins or protein fragments. They do not introduce the entire organism, making them safer. The journey begins by identifying the gene that encodes the important protein from the pathogen. Once this gene is isolated, it is inserted into a suitable host cell system. This could be bacteria, yeast, insect cells, or mammalian cells. These host cells act like tiny factories, producing large amounts of the desired protein. The first step in production involves inserting the DNA sequence that codes for the pathogen's protein into a plasmid or viral vector. This recombinant DNA is then introduced into the host cells. The host cells utilize their own machinery to transcribe and translate the gene into the target protein. After a period of growth, the protein is harvested from the culture, purified, and then formulated into a vaccine. Since only a specific protein subunit is used, these vaccines cannot cause disease. However, they still stimulate the immune system to recognize and respond to the pathogen if it is encountered later. Different host systems have their own benefits. Bacterial systems are quick and cost-effective but may not fold proteins correctly. Yeast and insect cell systems, like the Baculovirus SF9 system, can produce more complex proteins and virus-like particles that resemble the structure of the pathogen. Mammalian cells, such as Chinese hamster ovary cells, create proteins with human-like modifications but are more expensive and complicated to scale up. From the immune system's viewpoint, the purified proteins or virus-like particles act as antigens. They activate immune cells, leading to the production of antibodies and memory cells without the risk of infection. This targeted approach helps the immune system prepare defenses specifically against key parts of the pathogen. In real-world applications, recombinant subunit vaccines are used for diseases such as hepatitis B and human papillomavirus. The use of recombinant DNA technology allows for safer vaccines that effectively focus the immune response, reducing side effects while providing strong immunity. So, next time you hear about a vaccine, remember the science behind subunit vaccines and how they are made with recombinant DNA. They are a remarkable example of how we can harness technology to protect our health.